I'm going to be talking to you a little bit about things that have been hinted on as we've been going through this conference and looking at lutein in the brain tissue and what it could be doing there. I'd also want to acknowledge Emily Mon, who's my graduate student. And yesterday I learned on our talk that Isaac Newton, I think it was said that he stands on the shoulder of giants. Well, I stand on the shoulders of graduate students. They really do a lot of the work that's pretty important, and that's why I'm here today, and I'm very thankful for that. So before I get into some of the work that Emily and I, and in collaboration with John Erdman in Illinois and Martha, who you just heard from, before I get into some of the work that we've been doing, I wanted to give you some background on why we're doing this. And so I'm going to start out with some work that we did with Rohini Viswanathan a while ago, where we looked at macular pigment and a variety of measures of cognition. And we've been hearing about this at this meeting as well. Our lab has found it. John Nolan's lab and Billy Hammond's lab have found that there are these relationships between cognitive function and macular pigment levels. And what's really nice about this is that we're finding this in these very different populations. My population that we looked at were mostly Caucasian, about a third of them were African American, people with mild cognitive impairment, and the Irish population. So it's nice to see that confirmatory data on macular pigment being related to cognitive function. Something else that we've been hearing about is this relationship between macular pigment and the brain. Is it a biomarker of what's in the brain? Is it going to be a useful tool? Because we can't dig into the brain for a lot of these studies to find out, how much lutein or zeaxanthin is. So can we use this pretty easy device to measure macular pigment as an idea of what's going on in the brain? And this is work that I also did with Rohini Viswanathan. This is actually human data where we had matched retina with brain tissue. And you see this gorgeous correlation. So that we also did this work in monkeys with Martha, the help of Martha, where we found similar correlations in different brain regions. So this is a nice thing. Now we have this tool, and that helps us understand these observations that we've been seeing among these different groups of macular pigment with cognitive function. So this is something else that was talked about. Neil mentioned about, finding these carotenoids in brain, and what we find when we have matched serum with brain tissue is just like in the macula. We talked about lutein getting preferentially taken up into the macula along with zeaxanthin. What we see here is the same thing. So here's matched serum, and when you measure serum carotenoids, you're really getting an indicator of what people are eating. So in this population, they're eating a lot of beta-carotene. Beta-carotene was the major carotenoid in their diet. But with their matched brain tissue, look at that. It's lutein. So lutein, this tells us that lutein's getting preferentially taken up into the brain tissue when compared to these other carotenoids. So is that important? Is that something that we want? Do we want more lutein in our brain? Well, maybe we do, because when we did this study in older adults who had agreed to donate their brains upon death, we had this opportunity to actually measure carotenoids in brain tissue and match it with cognitive testing that was done within 6 months before death. So that's an amazing opportunity, and we took it. And what we found was we measured all the carotenoids. We used our C30 column, and we got them all. And the only one that we found that was related to this measure of dementia, and we looked at scales 1 through 3, and it goes through 7, where 4 through 7 are degrees of dementia. And what we heard earlier is once you get to Alzheimer's, it's really not as much that nutrition can do. Really, the target population is when you're normally cognitively intact or some mild cognitive impairment. So that's where we focus. And among all the carotenoids, lutein was the one that was related to this scale. We looked at cryptoxanthin, and Neil talked about how you get about the same amount of cryptoxanthin in brain tissues, but we didn't find that with cryptoxanthin. We didn't find it with beta-carotene. We didn't find it with any of the carotenoids. We didn't find it with alpha-tocopherol, which is really interesting because there's a lot more there, and it is an antioxidant. So we also looked at other measures of cognitive function, including the mini mental state exam, measures of verbal fluency, and so on. And again, lutein was the one that was pretty much consistently finding this relationship. So maybe there's something going on. And so lastly, what we did, and this, I think, study was mentioned, we actually did an intervention with lutein, and we found that lutein, along with plus or minus the DHA, which has been implicated in cognitive function, actually, these older women improved. So here we had all these little pieces of data to say maybe lutein's doing something. A lot of it's observational. We have our intervention trials. But what we really don't know is, what is it doing? If it's there, what is it doing? So that's what we're going to try and figure out, or hopefully we'll get some answers for you. So we're looking at mechanism now. And so what we thought we would do is look at lutein isolated in neuronal, mitochondrial, myelin, and nuclear membranes from the brain regions that are controlling cognition, and see if those concentrations in membrane is going to be associated with measures of brain cell viability. So we've done these measures in whole brain tissue, but now we're going to go down in on a subcellular level to see where is it, and does that help us understand what is it doing. So that's what we did, and this is in collaboration with Martha. She's sending us some monkey tissues, and John Erdman's group as well is helping us along with this project. And we're looking at adult monkeys where we're getting different brain regions. And what we're doing is we have some monkeys that are fed the standard chow, but because we're going to be looking at relationships, we really want to get a range of values. So we have monkeys that are going to be supplemented with lutein, and now we have completed 2 lutein supplement monkeys, and very soon we're going to be starting 2 more, so we'll be able to add to our numbers here. And so what we're doing is a lot. And so I'm not going to be talking about the data that we haven't got yet, because that's why I won't talk about it, but we're going to look at gene regulation and how levels of lutein in the membrane is going to be related to that. MAP kinase, to look at enzyme activation. It's related to apoptosis. And we're measuring measures of DNA oxidation in the nuclear and mitochondria. But what I will be talking to you about today is what we're finding in those membranes. So what our analysis is doing is we're taking that whole brain tissue and we're aliquoting it into various aliquots so we can do these various measures. And we're going through a sequential isolation of these membranes, doing a lipid extract for the carotenoids, measuring them by HPLC. But because of my interest in the fatty acids as well, because I'll be talking about how I think lutein and DHA are working together, we're on these same lipid extracts. We're measuring the fatty acids and cholesterol as well. We get that. Our data, we're going to be expressing by protein. And then today I'll be talking about some of our first measures on biomarkers of cell viability, where I'm looking at the neuroprostanes. And those are interesting because of the oxidative products of DHA. So what we first found is the supplementation worked. So that was great. And so what you see in orange are 2 of our supplemented monkeys with lutein, and in blue is the chow fed. And you see there is an enrichment of lutein in the tissue, in the serum. Here's lutein, the trans, we got some cis, we got enrichment of the zeaxanthin, and it looks like the other carotenoids are going up as well. So that's serum data. And this is what? I think this is out of place, but anyway, this is what's in the brain. Oh, yes, this is right. Okay, so this is what's in the brain. So we looked at the prefrontal cortex, the cerebellum, and the striatum. And again, the orange is the lutein supplemented, and the blue is the prefrontal cortex. We got the cerebellum in red, and we got the striatum in green. And again, you see the supplementation worked. So in these adult monkeys, we were able to enrich these brain tissues with a lutein supplementation. So that was really terrific for us to see. But now we're looking at what the membrane content is on these various tissues. And this is a little bit busy for you, and I'll take it through you one step at a time. But again, it's to show you that we are finding lutein in these membranes. And we're looking for all the carotenoids. But in this membrane tissue, we're not finding any other carotenoid. Sometimes we see zeaxanthin. We never see beta-carotene. We never see lycopene, although levels were low in the brain. We see them in the brain, but we're not seeing them in the membrane. It's just lutein. The other thing I want to point out is in the brain tissue, we're not seeing any of the cis isomers of lutein either. In the serum, we're seeing it, but it seems like that cis isomer is not getting to the brain tissue. And that's been pretty consistent among all these brain studies we've been doing. We don't really see that cis isomer, which is interesting. So here's the different brain regions. Here are the various membranes, the nuclear, myelin, neuronal, mitochondrial. We're finding lutein. We're only finding lutein in these tissues. So what happens when we supplement with the lutein? So here's the prefrontal cortex in blue. And it doesn't look like a lot because of the scale. I wanted to put all the membranes on the same scale. But it increased by about 2 to 3 times in the myelin, in the neuronal tissue, in the mitochondria. So we were able to enrich the membrane in these 3 membranes with lutein supplementation. We didn't see it for the nuclear fraction, though. We did see it, though, in the cerebellum. So I'm not really sure what's going on there. Why is it just the nuclear membrane in the cerebellum that gets enriched with the lutein supplementation? But that, in part, is why I'm here, because I want to pick your brains to help me understand that. So that would be very helpful if you could help me think about why we have this differentiation of lutein enrichment among the brain membranes, among the brain regions. So here's what we have in the striatum where the myelin's winning. The myelin is the one that really gets an enrichment of lutein with supplementation. So, the reason I put this one here is because that was expressed by milligram by protein in the denominator, and that was always been a question in our discussions when we're looking at these data, because when you look at these different brain regions, when you look at these different brain membranes, the composition can change quite a bit. So the question was, what's the best way to express these data? So this is looking at those data, looking at it by cholesterol in those membranes. And really the pattern doesn't change that much, but that's something that we're going to be considering when we look at all these various associations. So, for what I conclude from these first studies is that there's preferential uptake of lutein into subcellular membranes among all the carotenoids. So we really are just finding lutein, sometimes zeaxanthin, and that could be important when we're thinking about membrane function. So that's interesting. We also found that among these subcellular membranes that they can be enriched with lutein, but that seemed to be differential depending on where you're looking in the brain and what membranes in particular you're looking at. So is that related to function? That's the question now. Are these measures of lutein in these membranes going to be related to function? And because we're in the middle of this project, I can't really tell you too much about that. So hopefully I'll be invited back in 3 years to give you those results, where we can really have an understanding of what lutein is doing on that subcellular level. So now, to start thinking about that, I thought I'd get into a little bit about what we're finding as far as lutein and DHA in these membranes. And it makes sense to do this because just like with lutein, we're finding there's more information on these omega-3s and cognition. That has a longer history of a relationship. And so just like lutein, you're finding this DHA in those membranes. And just like lutein, you're finding this preferential appearance of DHA in the brain tissue. So these are data that we have from that centenarian study I described, looking at brain levels of carotenoids and cognition, where with those same brain tissues, we looked at the fatty acids. And what you see in their serum, yeah, among the polyunsaturated fatty acids, DHA, you know, there's a lot there, but look at what you find when you look at what's in the brain. So again, just like lutein, you have this preferential concentration of the DHA in brain tissue. So something that Julie, I think, mentioned yesterday is that you have this association of the omega-3s with the macular pigment. So this is a study that finds that. And that's interesting because if you think back to the beginning of my talk, macular pigment has been found in 3 studies now, or more, I think, after learning from posters, is that it's related to cognitive function. So the omega-3s, you know, may have a role in addition to its own role on cognition as far as enhancing that macular pigment. And indeed, we found that to be true in a supplementation study we did. I'm going to have to take you through this slide. So here's macular pigment. We're looking at differences from baseline with supplementation with lutein, DHA, or the combination. Differences from baseline with a 4-month intervention. And what we found with lutein, we got increases of macular pigment that were more eccentric. But what we found with DHA alone, so that's this line here, we found significant increases centrally. So somehow that DHA encouraged lutein to be taken up into the retina. So DHA and lutein seem to be hanging around together to help each other out, to help with this macular pigmentation. Are they helping with cognition? Well, in that same study, we also looked at cognitive measures, and I told you that lutein alone improved a measure of cognition. And what we found is that combination of lutein and DHA did the best in terms of these measures. You know, in terms of learning, it improved in these older women in terms of short-term memory. So the combination, looking at lutein and DHA together, seems to make sense based on these kind of data. We also, to remind you, we also, you know, had these brains for which lutein was related to cognitive function, higher lutein, better cognition. We looked at these same brains for the fatty acids as well. And for the variety of cognitive measures, and I won't go through this long list, DHA levels in these brain tissues was the most consistently related to better cognition, all right? Lutein among the carotenoids, most consistently rated to cognition. Same thing for DHA. So that's neat. And so what we found when we took those data and looked at the interaction, we found that the interaction between the brain concentrations of lutein and DHA was dependent on each other. So that relation each had to each other was dependent on each other. So perhaps they are working together. So given all that, given those different kinds of data suggesting that they're both important, they may be working together, that's part of this project as well. So we are looking at these membranes. These same membranes are being looked at for the fatty acids, and these are some of those preliminary data. So just like with lutein, we're having some differential uptake of the DHA I'm looking at here, differential composition of these membranes, depending on where you look, whether it be the myelin, the nuclear, the neuronal, or the mitochondrial membranes. And to remind you, it's the frontal cortex, the hippocampus for this slide, in the striatum. And our preliminary look at this is to find that there is a relationship between lutein and DHA concentrations in the subcellular membranes. However, it depends on where you're looking. So in the neuronal tissue, there was a significant inverse relationship, where in the mitochondria it's trending towards a positive relationship. So what does that mean? I don't know. But it might give us a hint to how it's functioning, and is lutein functioning differently in these tissues. So these are some of our measures on the neuroprostanes and the isoprostanes, which we heard about earlier. And the neuroprostanes are going to be your oxidative products of DHA, whereas the isoprostanes are your arachidonic acid. And when we look at these tissues as far as looking at the relationship between the neuroprostanes and the lutein, we find a significant inverse relationship, which is what you would expect. If lutein is acting to protect the DHA, you're going to see less oxidative product. And we see that same relationship with the isoprostanes, another oxidative product. Now, when we looked at, we have vitamin E data on these same tissues because we can measure that. We don't see it with the neuroprostanes and the vitamin E, so suggesting that lutein might have a unique role as far as protecting that DHA. And we also kind of divvied this data up in terms of young and older adults, suggesting that perhaps that relationship's going to change depending on age, where we find it to be stronger in the older adults, which kind of makes sense. You're going to have more of that oxidation going on. You're going to have a bigger range of values to be working with. It's going to be easier to measure these correlations. And so what I told you was we got macular pigment. It's going to be it's related to lutein and DHA status. Lutein supplementation plus minus that DHA improves cognitive function. We're finding them both selectively in that subcellular membrane, and that brain levels of each of them are related to better cognition, and that association is dependent on each other as far as what we looked at statistically, and that these concentrations between the two have been related in some of these subcellular membranes, although it depends on where you're looking. And that lutein is related to decreased oxidation of DHA in the brain, which is a hint to a mechanism by which it may be helpful. So how I want to end up is I gave you all this information. What does it mean? Is it important? What should we do with it? Maybe we should be thinking about dietary recommendations for DHA and lutein, because we talked about this yesterday. If you eat them, you don't die, right? You don't get a specific deficiency. But we might want to be thinking about optimal health now. And there are recommendations for the omega-3s, although they're a little bit varied. And there really is no recommendation for lutein right now. Around 6 milligrams, 6 to 10 is where we think is important for eye health. There's really nothing that we've been looking at as far as cognitive health, but really these discussions should be beginning, because we're seeing these data accumulate that really point towards the same thing, that DHA, lutein's important if we want to maintain neural health as we're aging. So here's a problem. We got these kind of ideas of targets we should get at, but at least in the United States, we're not there. You know, most people are below what we think is important. And when you look at the NHANES data, it's below less than 100 milligrams a day of the DHA and less than 2 milligrams of lutein. So there is a gap. So we really need to provide guidance for people to close that gap. So with that, I'm on the yellow still. So that's really nice. I have time to thank all these people who have helped me, and for some reason, something is missing that I thought I put in today, because it looks like, anyway, I'm sorry. I had acknowledgments for the funders. So the Center for Nutrition, Learning, and Memory, very important for the monkey studies. Abbott Nutrition, very important for some of the earlier work I presented. And DHA helped fund the fatty acid work in the membrane. So I really want to thank them and my collaborators who helped me get all these numbers to you to present and hopefully spark you to think about the importance of these bioactives.
So we have time for a few questions.
Great talk, Liz, about this dietary recommendations. Is it realistic, 6 milligram by diet for lutein?
Oh, it's easy. I do it all the time. Okay, so you buy, in the United States, you can get frozen spinach, 10-ounce package. It'll last you the week, you're fine. That's $2. And so Randy and I did a study a long time ago where they ate 60 grams of cooked spinach. That was 10 milligrams. That's that much spinach. It's nothing. It's simple. But the problem is no one's doing it. So that's why I had this conversation with Torbert yesterday. It's like, get that corn on the market, then we can help. People don't have to make too many dietary changes. They're eating their corn-based breakfast cereals. Maybe we can do it that way as well by really biofortifying the foods that we are eating. Because they're not eating green leafy vegetables, they're eating corn.
Just a kind of an add-on to that. The suggestion that it's easy, it probably is if we go to your kitchen for dinner. But the reality is people don't comply to that. And I also think, and I think they should, and I think we should have recommendations to try and get people there. So I agree with you. And then we have to take into consideration what George Britton was describing yesterday about how you prepare it and all those factors. And so I just think it's challenging. Yeah. We all know the issues with compliance to what we should be doing and changing habits and how we go about that, I think, is critical.
Right. And when I kind of played with the numbers, going through the dietary guidelines and what they're recommending in terms of fruits and vegetable servings a day, you can do it if you follow the dietary guidelines. You know, it's not like you're eating a bushel of spinach every day. It's not that hard.
Liz, I know you have a lot to do, but this is Stuart here. I know you have a lot to do, but there's also a folic acid DHA axis that is highly significant, almost R equal 0.6, very significantly related. So maybe if we used all three, lutein, DHA, and folic acid, we might even get a better result. Right. I don't know if you've looked at that or not, but that's
No, but I am interested. I'm lucky enough to work where I work because we all have our favorite nutrients, and there is a Jakob Selab who's in the B vitamin lab, has an interest in cognitive function, and we've been having those discussions as far as the importance of those B vitamins, particularly for the elderly. I work at a center for the elderly, and they are compromised from some of these important nutrients for cognition.
Okay.
Thank you very much for a really interesting presentation. I'm here. The other people have suggested that DHA can cause an increase in HDL, circulating HDL, and we've had a discussion about how these different carotenoids might be delivered. And I wondered whether you'd looked at whether HDL was being affected in your systems.
I'm glad you brought that up because usually when I have a lot to say about this, I talk about how lutein and DHA are working together. One being, DHA is huge, it's highly oxidizable, lutein is an antioxidant, you find them in the membrane together, but that's another point I make because lutein, the only way it gets anywhere is riding on a lipoprotein, and about 65 to 75% of the lutein you measure in blood is going to be on HDL. So that certainly is going to be a factor. And that's why I think, DHA can be affecting the transport to tissue. So by measuring total serum, that's good, you can do that, but it's really where is it in that serum? How is it distributed among those lipoproteins? And if it's on that HDL, I think that makes a difference. And I want to quickly bring up a study we just completed with avocados. And we fed these older adults 1 avocado a day for 6 months and measured cognitive function. In avocados, there's not a lot of lutein there. It's like 0.5 milligram. That's teeny tiny. But that, in an avocado, it just slides right in. And it slides right in, but not only into the circulation, but when we did our rough calculation comparing it to a lutein supplement, considering duration, dose, it was about 4 times better than a lutein supplement in terms of macular pigmentation. These people increased in their macular pigment, and that increase in macular pigment was related to better cognitive function. So I like to talk about foods as our vehicles, and it's the fats in avocados that I think made a difference, and that difference was on its transport into the neural tissue.
Okay, well, this is going to conclude this section. Everyone go and have a very nice lunch.